So without further ado, I am going to introduce Dr. Vignesh Kasinath. And he is a new professor at University of Colorado Boulder, just started in January, and it sounds like his lab is up and running, so that's really exciting. I'm sure if you're looking for postdocs and jobs, you can reach out. But he came from Dr. Ava Nogales' lab, where he was a Helen Hay Whitney postdoc fellow, and he has a Pathway to Independence Award, and he's published a number of wonderful papers, especially more recently on PRC2, for example, really making great strides in how we can understand how our chromatin modifiers look and the structures and their mechanisms. So it's really fantastic. And I'm really excited to hear his seminar today. I will stop rambling and give the floor to Vignesh. Thank you so much.
Thank you for this opportunity to speak at this series. I've been attending quite a few talks, not all of them for time constraints, but a lot of them. I think it's a great series, especially the Discord server as well. We were talking about it. I would encourage everyone to join the Discord as well. So my talk today is about structural insights into the recruitment and activation of PRC2. But I'm going to divide my talk in two aspects. I don't know if I'll have the time to get to the second one, which is more our understanding how can we look at complexes which organize entire chromosomes, especially ones that we cannot isolate to study. But for the most part, I'll be talking about this complex called PRC2, which is fundamentally important for cellular differentiation and maintaining cell identity. And it does so by regulating the architecture of the genome, essentially by maintaining a balance between actively transcribing regions and transcription repression regions, or repressed transcription. So as you guys are, no need to introduce this aspect to you, but our DNA is organized into extremely complex but defined structures, with the nucleosome being a fundamental unit of such a structure. The nucleosome is a heterooctamer with 2 copies of 4 histones, H3, H4, H2A, and H2B, with DNA wrapped around it. Post-translation modifications of the histone tail are critical nodes for transcription regulation. But for the purposes of this talk, I'm only going to focus on the post-translation modifications of the histone H3 tail, more specifically, the lysines in the histone H3 tail. There are several lysines. I'm going to talk about the 4 most important ones in my mind, which is 4, K4, K9, K27, and K36. Some of these will kind of repetitively appear throughout the talk, so I'm going to introduce them now. So each of these lysines can either be acetylated or methylated. Acetylation, for example, of K9 or K27 mark genes for active or mark regions of active transcription. Similarly, methylation, monodi or trimethylation of K4 and K36 also mark regions for active transcription. K4 trimethylation is particularly found in regions of gene promoters, whereas K36 is found in the genome bodies. However, K9 and K27 methylation mark them for facultative heterochromatin or heterochromatin boundaries. Specifically, there are a whole set of complexes which are highly spatially and temporally regulated to carry out this reaction. These reactions are covalent but reversible modifications. And specifically, there are a whole group of enzymes which carry out the methyltransferase reactions or acetyltransferase. Similarly, there's a whole group of enzymes that carry out the demethylase reactions. Of particular note is the methylation of K27. And the enzyme that carries out this reaction is called EZH2. And EZH2 is part of PRC2. So PRC2 itself is actually part of much larger complex. It's called the polycomb repressive complex, which contains 2 other members, PRC1 and PRDUB. PRDUB is a deubiquitinase. PRC1 is actually the most diverse family in this complex, which is an E3 ubiquitin ligase. And it also has a secondary role in compacting chromatin. I won't talk too much about PRC1 today, except for you guys to know that this enzyme is critically responsible for depositing monoubiquitin on histone H2A at lysine 119. This is an aspect that will also come about throughout the talk. But for the most part, I'm going to focus on PRC2, specifically PRC2 with these 2 cofactors, JARED2 and AEBP2. So PRC2 itself actually exists in several different flavors. The 2 most important ones are PRC2.1, which has a specific subset of cofactors, and PRC2.2, which has JARED2 and AEBP2. So I'm going to focus on PRC2.2 today. So what is PRC2 and what does it do? So the primary function of PRC2, which contains 4 core enzymes, you don't need to remember the names, is mono, di, and trimethylation of histone H3 at lysine 27. And by trimethylating histone H3 at lysine 27, it marks genes for transcription repression. But it has a secondary another secondary function which is also equally important, which is once this trimethylation mark is deposited on histone H3 lysine 27, PRC2 also spreads this repressive mark to neighboring chromatin regions, establishing heterochromatin boundaries. So besides these, I mentioned that I'm going to talk about cofactors JARED2 and AEBP2. So these two cofactors are fundamentally important during embryonic stem cell differentiation, and they play they play a number of roles. Most important among them being is regulating the activity of PRC2, recruiting PRC2 to the genomic loci, specific genomic loci, and several others. Because of the critical role that PRC2 plays in establishing and maintaining transcriptional homeostasis, especially maintaining the transcriptional program through differentiation, it is often found mutated in almost every cancer, every type of cancer, and different types of mutations. But fundamentally, we would still like to understand how PRC2 dysregulation results in these cancers are in fact the hallmark of many of these cancers. And to look at this, in my mind, there are three fundamental questions one needs to answer for any chromatin modifier, which is how is the activity of a chromatin modifier regulated? And in this case, how does PRC2 deposit H3K27 trimethylation? And what are the factors that regulate this initial deposition? Secondly, how does the chromatin modifier get activated on the chromatin or at the chromatin? In this case, how does PRC2 spread this repressive H3K27 trimethylation mark to neighboring chromatin regions? And the last, probably the most important, is how does PRC2 get to the right genomic loci in the first place? And what are the factors that regulate it? So I'm going to give you the most important tidbits of the first two, and I'm going to focus more on the last aspect today, which is our most recent study. And to look at this, over the last 2 decades, there have been several structural as well as biochemical studies. A lot of these studies have initially focused on individual domains of PRC2, with the first full structure coming from Eva Logales' lab in 2012, which used negative stain electron microscopy to define how the architecture of these enzymes look like. But still, there had been no high resolution structures of PRC2, especially towards understanding the role of JARED2 and AEBP2, and how do they regulate PRC2 activity. So we decided to use cryo-electron microscopy for this. And I'm going to give you a 2-slide primer for cryo-EM. It's going to be very short and concise. It's not going to be detailed, but just to get everyone oriented in the same direction. So what happens in cryo-EM or cryo-electron microscopy? So we have our isolated protein molecules of interest, and we plunge freeze them into a frozen hydrated state such that these molecules are randomly oriented in 3D. We then image them in an electron microscope, which essentially generates 2-dimensional projections of these randomly 3-dimensionally oriented objects, in our cases, protein molecules. So this is an example of a typical, but a really good cryo-EM image where you can easily see individual protein molecules. So we can pick these out. Once we pick them out, we can look at what the relative orientations are with respect to each other. We can align them to generate something called a 2D class average. Typically, in a sample, we can generate several such 2D class averages corresponding to the different views, which we can then put together to generate a 3-dimensional reconstruction or 3D structure, which we can model into. But the real power of cryo-EM is that we could generate multiple different conformational states in our sample. So essentially, if your complex has an ensemble of structural states, which we can visualize, cryo-EM should be able to discern each of those states. This has been applied to a lot of different systems over the years. So what did we learn? So the first question I'm going to talk about is how do these cofactors regulate PRC2 activity? So we looked at the structure of the core PRC2, which contains 4 enzymes together with these 2 cofactors. So we were able to define the complete architecture of this enzyme. I'm not going to go into the detail, except that I'm going to tell you guys that PRC2 is primarily organized into a catalytic top lobe and a regulatory bottom lobe. So, but specifically, how do these cofactors regulate the activity of PRC2? So what we found was both cofactors, surprisingly, mimic histone tails in their interaction with PRC2. So JARED2, for example, mimics the product of PRC2, which is H3K27 trimethylation. And it also interacts in the same region where H3K27 trimethylation is found to interact with PRC2. This mechanism is important because the interaction of PRC2 with either the JARED2, which is trimethylated, or the histone H3 trimethylation, which is the product of PRC2, is important for allosteric activation. And this has also been observed before from mass spec and biochemical studies. But what we also saw in our structure is, besides this trimethylated segment of JARED2, which binds the allosteric site, We also found the substrate, essentially the same segment, but without the trimethylation bound to the active site. So essentially, this light blue is the allosteric site and the dark blue is the catalytic site. So the substrate, which is unmethylated, is bound on the catalytic side, and the product, which is trimethylated, is bound on the allosteric side. So essentially, Jared2 serves both as an allosteric stimulator as well as a substrate for PRC2. Okay, so what about AEBP2? AEBP2, as it turns out, also does something similar, but it plays a more scaffolding role. It is anchored by its interaction with the regulatory lobe of PRC2, the bottom lobe, but it does so by mimicking unmodified H3K4 segment of the histone tail. H3K4, the unmodified segment of H3K4, has been previously shown to interact with one of the subunits in the regulatory lobe. So AEBP2 seems to be anchored through the same set of interactions through this lysine-arginine finger. But besides that, it also links the top catalytic lobe and the bottom regulatory lobe to stabilize the entire scaffold. So they both play completely different roles, but together, synergistically, they allosterically activate and stabilize PRC2. And this is important because PRC2 by itself is actually a really poor enzyme, which I'm going to get to in a minute. So the presence of Jared2, which is an allosteric activator, and the presence of AEBP2, which is a structural stabilizing scaffold, helps PRC2 initially deposit H3K27 mark, which is the transcriptionally repressive mark. So the next question is, okay, so now Jared2 and AEBP2 seem to mimic histone tails and help with the initial deposition. So how does PRC2 actually spread this mark? So we actually got some clues from our initial study as well, because Jared2 is both a substrate and an allosteric activator. And in our structure, we were able to see both the substrate form and the allosteric activator form bound to PRC2 simultaneously. So perhaps a similar mechanism can be used by PRC2 to bind the H3K27 trimethylation or recognize the H3K27 trimethylation mark and deposit a new trimethylation mark on the neighboring nucleosome. So could this be a mechanism? To look at this, another former colleague of mine who now has his own lab in the University of Cologne, he pioneered this method where he looked at PRC2 interacting with a dinucleosome, such that one of the nucleosome is a substrate, basically unmodified H3K27, and the neighboring nucleosome has trimethylated H3K27, which will serve as the allosteric activator. And what Simon saw was really interesting. In fact, it's a fascinating structure. PRC2 seems to be sandwiched between the two nucleosomes, and it's primarily interacting with the DNA, which is different from most chromatin modifiers, which interact with both the DNA as well as the acidic surface of the acidic patch of the histones. In addition, we also saw something weird. We only noticed half of PRC2, essentially only the top catalytic lobe, the bottom regulatory lobe is actually missing in our structure, which is kind of going to be the focus of the rest of my talk, and I'm going to get to it in a minute. But this essentially confirmed what we had observed for the case of Jared2 as well, where the nucleosome, which contains the trimethylation H3K27, was bound to the allosteric side, whereas a substrate nucleosome, which contains the unmodified H3K27, is threaded into the catalytic side. Okay. So this could be one of the plausible mechanisms. This may not be the only mechanism in which PRC2 is spreading H3K27, but it could be one of the plausible mechanisms. So, and this is classic in terms of a feed-forward mechanism where it recognizes its own product, gets activated, and deposits a new mark on the neighboring nucleosome. So the question at this point was, okay, so we know the cofactors regulate PRC2, and it does so by mimicking histone tails. We know PRC2 interacts simultaneously with a oligomeric nucleosome, in this case a dinucleosome, and employs this feed-forward mechanism. But how does PRC2 get to the right genomic loci in the first place? And what are the factors that could regulate it? And do cofactors play a role in this, as they have been hypothesized to? And there are several factors that have been known to play a role, like CpG DNA. In mammalian cells, polycomb target genes are enriched in CpG islands. RNA, non-coding RNA especially, has been thought to play a role in PRC2 recruitment. Besides these, PRC1, which is the other polycomb member, is also thought to play a role through its catalytic activity. So we wanted to look at two aspects of this problem. One is, okay, PRC1, which deposits monoubiquitin and H2A K119, is thought to play a role. So how does this play a role, and what role do cofactors play in this? There's been some discord between what role exactly H2A K119UB plays. Some argue that it could inhibit PRC2 activity. Some argue that it actually promotes PRC2 recruitment and activity. Similarly, on the flip side, active transcription marks such as H3K4 trimethylation and H3K36 trimethylation, which mark regions of active transcription, are thought to inhibit PRC2 core activity. This is important because one would not want PRC2 to act on regions of active transcription, because then it will aberrantly repress those regions, and that's not good. So this is so historically, this has been thought that active transcription markers inhibit PRC2 activity, and there's a feedback mechanism between PRC1 and PRC2 which could regulate PRC2 recruitment. But to look at this, we first wanted to get over this bottleneck of not visualizing the regulatory low. So I'm going to cut to the chase. We know why this is the case. This is because there's a fundamental problem with cryo-EM is when we make our samples for imaging, we have an extended air-water interface, which essentially is hydrophobic, and that can denature protein complexes. And this is exactly what is happening in the case of PRC2 when we look at it with nucleosomes. So how did we get over this problem? We tried several things. Some of them worked, some of them didn't, but I'm only going to highlight one such mechanism, one such process that worked. And this technique was actually pioneered by Dr. B.G. Hahn and Bob Glaser's lab in Berkeley. The method is called Affinity Grid Prep for EM. The idea is really simple. Okay, so the complexes, once they get exposed to the air-water interface, denature because it's hydrophobic. So can we now prevent these complexes from going to the air-water interface as much as possible? And the way we carried out this process for PRC2 nucleosome interactions is we put a biotin tag on the DNA of the nucleosome. And when our complex of PRC2 nucleosome forms, if we have a streptavidin monolayer, the biotin streptavidin interaction should keep the complex away from the air-water interface. So this actually ended up working really, really well. And this is probably a preferred go-to method now for polycomb, because most of these systems end up getting denatured at the air-water interface. So by utilizing this method, we looked at PRC2 bound to a mononucleosome, which contains H2A-K109UB as well as CpG DNA. And the structure was actually phenomenal, probably our best structure so far, because we saw in exquisite detail a lot of different things that we hadn't seen before, as well as some insights that we knew what would happen, but having the ability to visualize it kind of gave us new insights into how PRC2 is regulated. So I'm going to walk you through probably two important aspects of this complex so that kind of everyone is on the same page. The first is we were able to visualize the complete H3 tail and what are the factors that regulate the H3 tail threading into the catalytic site. So our model and our map was such high resolution that we were able to build a completely de novo most of the H3 tail. And one thing that we did see was we were able to identify all of the residues that have been shown to be mutated in several cancers and their importance in terms of threading the H3 tail into the catalytic site. In fact, we even identified some residues which do not directly interact with the H3 tail, but it was important for organizing the catalytic site so that the H3 tail can be bound in the right geometry in the site. Besides this, we also identified this new segment, which we call a bridge helix, because this particular helix is only visualized and only ordered when you have a nucleosome substrate. So something unique with PRC2. And this helix sits right at the tripartite interface between PRC2, histone tail, as well as the nucleosome DNA. And it's critical because it sits right on top of the H3 tail containing the amino acid H3K36. So one of the things that we are looking into is whether this helix is important for sensing the post-translational modification state of H3K36. Perhaps this is a mechanism that is important for regulating the activity in actively transcribing regions. So what about the cofactors and how do they interact with this H2A K109UB mark? And if at all they do. In fact, I'm showing you the structure because they do. Jared2 harbors a segment called ubiquitin interaction motif, which it's a sandwiched between one of the ubiquitins that is on H2A K109 as well as the histone acidic surface. In fact, Jared2 also harbors another lysine arginine rich segment which sits on top of the acidic surface. So one of the things that we did notice in this was if we don't have the H2A K109UB, we do not visualize this segment of Jared2 interacting with the nucleosome. Similarly, if we do not have this segment of Jared2, we don't see the ubiquitin ordered. So it seems like the presence of ubiquitin is necessary for Jared2 to interact with the nucleosome. And this is also the case on the flip side, on the other side of the nucleosome in the case of AEBP2, where we found that the first two of the three tandem zinc fingers of AEBP2 interact with both the histone acidic patch as well as the ubiquitin simultaneously. So this asymmetric interaction of both the cofactors with ubiquitin is important because in the absence of ubiquitin, we don't see either of these interactions. And similarly, in the absence of these regions within the cofactors, we do not see the ubiquitin ordered as well. So this tells us that the presence of H2A, K1 and UB is important to recruit PRC2 via JARED2 and AEBP2. This could be one of the contributing mechanisms, because in our structure, we also have CpG DNA sequences, and we see other regions of AEBP2 also interacting with that, which I'm not going to go into. So one of the interesting things that we did observe was this tandem zinc finger of AEBP2 has previously been shown to interact with CpG DNA. So in our structure, what we do see is perhaps these zinc fingers play dual roles. It could also interact with CpG DNA, but in the presence of ubiquitin, especially the ubiquitin on H2AK119UB, these zinc fingers primarily interact with ubiquitin to stabilize the PRC2 nucleosome interaction. So it's all good now that both cofactors interact with this H2AK119UB specifically. But does this interaction also result in regulating the activity of PRC2? Because not only do they recruit, but maybe perhaps they also activate PRC2 on the specific genomic loci, which is something that we looked into next. So besides this, we also wanted to understand whether H3K4 trimethylation and K36 trimethylation similarly affect the activity of PRC2 in the presence of cofactors, which I think has not been looked into in that detail before. So what we found was similar to what had been observed before. The core PRC2 without cofactors is a very poor enzyme. So irrespective of what post-translation modification that exists on the nucleosome, the activity of core PRC2 is really bad, really poor. And in the presence of K4, K36, it's actually even worse. And this had been observed before. When we have AEBP2 in our structure, so remember AEBP2 plays the scaffolding role, it stabilizes the complex, but it doesn't activate the complex. We see something similar. The unmodified nucleosome, basically, I would say that the activity is still poor. There is still inhibition in the presence of K4 and K36, which we'll come to in a minute. So, however, in the presence of JARED2, which has this allosteric activation segment, the segment that gets trimethylated by PRC2, we see a dramatic increase in methyl transferase activity of PRC2. And there's no change when you have a ubiquitin on the nucleosome, because this segment of JARED2 does not contain the ubiquitin interaction segment. However, in the presence of K4 and K36, now what we observe is there's a higher activity on these nucleosomes as well, which is a little bit surprising because K4 and K36 had been shown to inhibit PRC2 activity. However, when we have both AEBP2 and JARED2 and different combinations of these, what we see is we still see partial activity on the K4 and K36 nucleosomes. However, in the presence of the complete AEBP2 and JARED2, which contains both the ubiquitin interaction segment as well as the allosteric activation segment, PRC2 in the presence of nucleosome containing the K119UB, H2A-K119UB, shows increased activity. So, but it's modest. It's not, I wouldn't say, I think the primary contribution is still from the allosteric activation, whereas the ubiquitin plays a modest role in activating PRC2 further. Perhaps it's more of a kinetic mechanism where the turnover is now, PRC2 binding to nucleosome and the turnover is now increased. So overall, the main conclusion for us from this was the allosteric activation by JARED2 is able to partially overcome H3K4 and K36 mediated inhibition, as well as the fact that there's a modest increase in activity in the presence of H2AK119UB, which primarily plays a recruitment role. So we asked ourselves, okay, now that we see this partial activity, what could be the mechanism of this partial activity, and why is there still inhibition in the presence of K4 and K36 trimethylation? So to do this, we looked at again PRC2 with nucleosome containing K4 and K36 trimethylation using cryo-EM. Surprisingly, what we did, so in this case, we're looking at K4 trimethylation containing nucleosomes. What we did see was we saw two conformational states. Again, goes back into this idea that cryo-EM can separate multiple conformation states. In one of the conformation states, we saw the H3 tail bound in pretty much identical fashion to what we had seen before. However, in the second state, we saw that there was no H3 tail bound. So the nucleosome is still bound to PRC2 in the same geometry. However, the histone H3 tail is no longer in the catalytic site. So there has been a lot of work looking at how post-translation modifications of histone H3 tails particularly result in increased tail dynamics. So which is our favored mechanism right now, in the sense, maybe the presence of the H3K4 trimethylation results in increased tail dynamics that results in these two different conformations. So if the H3 tail is no longer in the catalytic site, it's not going to be acted upon by PRC2. So which is probably one of the reasons why we see this partial inhibition. Similar studies are also being carried out now for H3K36, which, if you remember, sits right below this helix that's at the interface of PRC2 and nucleosome. So overall, I would say, I think over the last 2 decades, there's been a ton of structural studies. Now these studies are getting more and more complex. But so far, I've only talked about structures of PRC2. And since the pandemic hit, there's actually been a lot more structures that have also come out. Now looking at different PRC2 complexes, I didn't mention anything about this EZH1, which is a paralog of EZH2. I also didn't mention anything about PRC2.1, which is another subset of PRC2 complexes, which has different set of cofactors. In fact, there's been not a lot of structures on this as well, which people are looking into. But overall, the theme that I've observed is, besides this complex interaction with nucleosome, now we're seeing more and more oligomeric states of these chromatin modifiers interacting with nucleosomes. The functional relevance of this is still something I think people are actively investigating, including us, but I think that's the general trend of the field now, looking at what are the functional relevance of these oligomeric states of PRC2 interacting with nucleosomes. So let's just summarize this part. So in the first part, I talked about how cofactors mimic histone tails, and they mimic Jared2 is able to mimic both a substrate as well as an allosteric activator to stimulate PRC2. ABP2 mimics an unmodified H3K4 tail to stabilize the complex. And they both synergistically activate PRC2 because the complex is now much more active. And this is important for the initial H3K27 deposition on the nucleosome. But once the H3K27 trimethylation is deposited, PRC2 can now recognize this mark and act on neighboring nucleosomes to spread this mark. And both these cofactors, Jared2 and ABP2, recognize the product of PRC1, the other polycomb member, in recruiting PRC2 to the right genomic loci. In fact, recent studies have now showed that the catalytic activity of PRC1, especially its E3 ubiquitin ligase activity in depositing or marking regions with H2A, K11, and UB, is important for PRC2 activity because it specifies which targets PRC2 needs to be active on. However, there's still a lot of questions that us and other people are interested in. For example, how are PRC2 complexes which do not contain ABP2 and Jared2 regulated? As well as how does RNA regulate PRC2 activity, which I didn't mention at all in this talk? As well as how other post-translational modifications, I talked about H2A-K11 and UB and H3K27 trimethylation. What about phosphorylation? What about K4 and K36 trimethylation? Mechanistically, how do they still regulate PRC2 activity? These are studies that are still being carried out. I think with that, I'll stop. I'm happy to take questions on other aspects. I'm not going to go into the second part. I think that might push it a little bit, but I would like to thank Eva because a lot of the last story was done in Berkeley while I was still a postdoc. And I was helped by two amazing undergrads. Curtis is right now at Johns Hopkins. Jennifer is right now at MIT. And of course, this is my Twitter, my lab website. I'm also looking for postdocs. And this is my current group right now. Feel free to write to me if you guys have any questions. Thank you.
That was fantastic. Really great to see how PRC2 mechanisms you've really added so much more to our understanding of that. So that's really great. So we have a couple questions already in the Q&A box. So the first one we have from Sivash Kurdistani. And he says, great talk and discoveries. I agree. Does binding of PRC2 to one H3 tail prevent binding to the other tail in the same nucleosome? Or is there sufficient room for two PRC2 complexes to bind simultaneously to the two H3 tails of a nucleosome? And the second question is SAM required for binding of PRC2 from the histone tail?
Yeah, so I mean, this is actually what I was hinting at earlier. There's an oligomeric state of, like, basically a higher order state of interaction of PRC2 with chromatin. And so we initially observed two PRC2 molecules interacting with the same nucleosome. In fact, I didn't talk about this, but this is part of the same study. This is another confirmation that we were able to pull out from the cryo-EM analysis. In this confirmation, each H3 tail is bound by an individual PRC2. And this theme has also been seen similarly for the case of PRC2 EZH1, which I think came out earlier this year as well, where they found that each H3 tail, based on my understanding, each H3 tail is now interacting with a specific PRC2 EZH1 molecule. But the distinction between these is that in our case, the two PRC2s are actually not interacting with each other. Whereas in the PRC2 EZH1 case, there's actually a domain swap dimer that forms. We see this domain swap dimer. We also see this domain swap dimer, but we actually see several different domain swap dimers which I don't want to talk about it too much at this point, but we do see similar configurations for PRC2, EZH2 as well.
Cool.
And is SAM required for binding PRC2? Yeah, I mean, in all our binding assays, it doesn't really matter if you have SAH or SAM in terms of binding to nucleosome. But in all our structural studies, we do have SAH as part of the structure. In fact, we do actually see the SAH bound to the molecule. But I don't think it's critical. SAM is required for activity, but I don't think it's critical for binding.
So for the next question is from Gowrie Deek. How did you pinpoint the relevant PRC2 binding segments of JARED2? Do you have any hypotheses about the role of the JMJC and arid domains of JARED2, since the JMJC domains of the JARED1A enzymes bind H3K4 marks?
Okay, so the first question, how did I pinpoint the segments of JARED2? This was a complicated aspect that I didn't get into, because we only saw cumulatively about 40 amino acids of JARED2 bound to PRC2 in our first study, and JARED2 is 1,271 amino acids. And a lot of it is unstructured, and probably just like how in the case of the nucleosome, some parts may become ordered given the right substrate and the right binding partner. But the way we went about it was our model was sufficiently high resolution to model the side chains directly. But in some cases, we did validate, in fact, not in some cases, in all cases, we did validate it with cross-link mass spec. So we knew exactly what segments of JARED2 were cross-linking to segments of, let's say, SUSY12 or EZH2, or even AEBP2 for that matter. So we were able to validate all our modeling with cross-link mass spec. And we also had structures of PRC2 without the specific segments of JARED2, which we were able to confirm if we lack the density in our model or in our reconstruction, then we know that it likely belongs to that segment. So we had multiple lines of corroboratory evidence. And in the second question, so do you have any hypothesis about the role of JMJC and arid domains? So JARED2 is a little bit unique because it's actually, even though it does have a demethylase domain in the C terminus, it's actually not a functioning demethylase because it lacks a critical histidine in that catalytic site of the demethylase domain. However, the arid domain as well as some of the other domains are thought to be important for binding AT-rich segment. However, we haven't really looked into it because, and it's also thought maybe it also could play a role in interacting with RNA. Yeah, there's a lot of uncertainty. I mean, there has to be more studies on this, but my suspicion is even though polycomb target genes are enriched in CpG islands, perhaps the C-terminal domain of JARED2, which contains arid, JMJC, and some other segments, may be important for binding RNA or maybe other segments of DNA in recruiting, because recruitment is a very complex process. I mean, it's not just the H2A, K1 and UB, or not just the CpG DNA that's going to specify recruitment. There are probably other factors that play a role in this as well.
We have a lot of questions. So those of you who are, oh, and I forgot to mention verbally, if you'd like to stay for the coffee chat, feel free to write in the chat box that you would like to do that. And we have some great people joining us for that. I'm going to skip their questions and we can ask those during the coffee chat and move on to those who are not staying. So Joy Choi asks, were you able to tell whether PRC2 indeed prefers CpG island / CpG rich sequences over other DNA, for example, 601 sequence from your structure?
Yeah, so I don't think we have a structure yet of PRC2 bound to nucleosome which does not have a CpG DNA overhang. But just based on looking at our structure, the interaction regions with the CpG DNA are specifically the overhangs are minimal. There's a segment of AEBP2 which contains these, maybe I can point it out here, is this small red blob right here of density that contains this lysine arginine rich segment, which has been shown by other labs, including Danny Reinberg's lab, to be important, and Tom Check's group, I think, to be important for binding CpG DNA, including the zinc fingers that I mentioned. But I would say the interaction interface specifically with the CpG DNA is minimal compared to the primary interface where the catalytic site, as well as this new bridge helix that we see now, interacting with the nucleosome DNA. That is the dominant interface. So Yeah, I wouldn't. It may be important in the context of in vivo studies because there's a lot of elements that pretty much recruit PRC2. But just from this isolated structure, it's kind of impossible for us to say if CpG DNA is a determinant in recruiting PRC2. It's probably one of the factors, but may not be the most important factor.
We have a lot of questions. That's fantastic. I'm going to ask one more just for the sake of time. And so we'll ask from Allison Swain. She asks, PRC2 activity is measured by SAH production in your work. I'm wondering how this correlates with H3K27 trimethylation, as that seems to be the functional output for PRC2. Do you know if H3K27 mono and trimethylation differ under different conditions?
So the assay that I showed, you're right, is a cumulative methyltransferase assay. It essentially measures mono, di, and tri. So, but I didn't show you the Western blot because that's in our paper. You guys can take a look. But we did profile the H3K27 trimethylation specifically because that's probably more important, because we wanted to make sure in the presence of these post-translation marks, we still do detect, or maybe the lack of the trimethylation product of K27. However, we did not do Western blots for the mono and di to extensively profile this. I think there have been other studies that have done mono and di profiles using Western blots for core PRC2, but I don't know if there are extensive studies looking at both cofactors with Western blot analysis for mono and di trimethylation of H3K27. To give you some context, you will find maximal amount of dimethylation usually for when PRC2 acts on nucleosomes. So dimethylation is the most abundant product, and then you get trimethylation, because going from di to tri is actually probably the slowest step, because the bulkier group of dimethylation when it's in the catalytic side reduces. I think there is kinetic effects and maybe thermodynamic barriers that PRC2 has to overcome to do the trimethylation. So, but to answer your question, we did not do the mono and di quantitation using Western blot, but we did profile for the trimethylation, which I didn't show.
Cool. And I think with that, we should probably wrap it up and move on to the coffee chat. But thank you all for joining us today. And thank you so much, Vignesh, for a fantastic seminar. Really excited to see what your lab does too.